Absorption refers to the transport of drugs across the membranes of mucosal cells in the gastrointestinal tract. A drug enters the gastrointestinal tract through the mouth and moves into the stomach through the esophagus. Once in the stomach, the drug dissolves. Acidic drugs are absorbed in the stomach because its acidic contents cause the acidic drugs to remain unionized, uncharged, for easy absorption. Alkaline drugs, which cannot be absorbed in the acidic stomach, pass through the pyloric sphincter into the first part of the small intestine and are absorbed there. Once absorbed in the blood, the drug molecules travel through the liver via the portal system. Some drugs are immediately metabolized in the liver. This is called first-pass metabolism. Others may be secreted back into the small intestine in the bile in what is known as enterohepatic cycling. Other parts of the oral dosage may be absorbed in the large intestine or fecally excreted. All of the drug dosage may not be absorbed. The drug molecules may bind to intestinal contents, which prevents their being passed through the membranes of the mucosal cells. Or they may be broken down by bacteria in the ileum and colon. Distribution is the process by which drug molecules enter the bloodstream, circulate through the body, and are transported to target sites of action. Distribution begins when drug molecules enter the blood from the liver, where they have been metabolized. The molecules then travel all over the body via the vascular and lymphatic systems. It is from the capillaries that the molecules are distributed to target organs and tissues, as well as to tissues where they will exert no action, for example, storage sites, sites of inactivation, and sites of excretion. The capillary wall is composed of a single layer of endothelial cells. The drug molecules diffuse across the endothelial cells of the capillary into the surrounding interstitial fluid and then into tissues. Because the drug molecules are delivered by the blood, the entry rate of a drug into a tissue depends on the rate of blood flow into the tissue mass, and the rate at which entry and exit of the drug molecules between blood and tissue equilibrates. Distribution may be uneven when there are differences in blood perfusion, tissue binding of the drug molecules, regional pH, and permeability of specific cell membranes. Drugs are eliminated through several organs, including the bowel, the skin, the lungs, and most often the kidneys, from which they are eliminated in the urine. Drugs are eliminated unchanged or as metabolites. Each kidney is composed of more than 1 million closely packed functional units called nephrons, which comprise a glomerulus and a tubular structure. Renal excretion of drugs and metabolites involves three processes: glomerular filtration, passive tubular reabsorption, and active tubular secretion. Most drugs or their metabolites are excreted in the urine by glomerular filtration. First, the drug molecules are filtered through the glomerulus. And next, the filtrate, the substances that have been filtered, move into the proximal tubule. Some tubular reabsorption may occur in the proximal tubule as the filtrate moves through. The drug or its metabolites in the proximal tubule may be actively secreted. Compounds that are secreted usually also undergo glomerular filtration. Renal excretion then is a combination of both passive filtration and active excretion.